Hello, everyone, and thank you for joining us for the third and final presentation of our immuno-oncology series. Today's live broadcast, Advancing Precision Immunotherapy Through Next-Generation Sequencing of T-Cell Receptors, Part 2, Oncomine TCR Beta SR Assay, is presented by Dr. Timothy Looney, Staff Scientist, Bioinformatics, Thermo Fisher. Last month, Dr. Looney highlighted novel applications of long-amplicon TCR beta, TCRB sequencing. Today, we introduce a newly launched short-amplicon TCRB sequencing assay designed to interrogate the TCRB CDR3 region from fresh or formalin-fixed paraffin-embedded FFPE samples. I am Alexis Krauss of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit thermofisher.com. Now, let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Answer a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Dr. Timothy Looney. Dr. Looney is an accomplished computational biologist and an immunologist with expertise in methods and applications of B and T-cell receptor sequencing, including biomarker discovery and assay development. He is currently a staff scientist and informatics lead for Immune Repertoire Sequencing with the Clinical Sequencing Division at Thermo Fisher Scientific. Dr. Looney's interests and specialties are centered around repertoire analysis for immuno-oncology, autoimmunity, and infectious disease translational research. For a complete biography on Dr. Looney, please visit the Biography tab at the top of your screen. Dr. Looney, you may now begin your presentation.
Great, thank you. Thank you for that nice introduction. It's a pleasure to be here to talk to you about our new TCR beta short read SR sequencing assay. And to get started, I want to give you an outline of the slides that I will present. I'll start by giving a brief introduction to TCR sequencing and the SR assay design, after which we'll switch to applications of the TCRB SR assay, which include interrogation of FFPE preserved samples and detection and tracking of rare clones for longitudinal analysis of clones of interest. And then in the third section, we will explore data addressing T-cell repertoire features of chronic cytomegalovirus infection. I think you'll find this interesting. So to get started, I want to remind everyone that our TCR sequencing assays are part of a broad portfolio of assays aimed to help you with your oncology research needs. And within the immuno-oncology solutions we provide, there are 4 assays. The Oncomine Immune Response Research Assay, which is a targeted gene expression profiling assay designed for interrogation of the tumor microenvironment. The Oncomine TCR Beta LR assay, long read. This is what we discussed in the previous webinar, and this is a wonderful assay for interrogating peripheral blood. The SR assay, as I mentioned, is the focus of this webinar, and then the Oncomine tumor mutation load assay is designed for quantifying tumor mutation from sequencing of tumor. And I'll mention that the strategy in the choice of these 4 assays is tasked with interrogating all aspects of the tumor immune interactions that are part of the cancer immunity cycle. So this ranges from profiling of tumor neoantigens through the Oncomine tumor mutation load assay, within the peripheral blood, detecting T-cell signatures of response to tumor neoantigen through the Oncomine TCR beta LR assay, and then within the tumor microenvironment, use of the TCR beta SR assay and immune response research assay, which are both compatible with FFPE preserved material. And these assays, we believe, help to address two critical questions in immuno-oncology. First, what are predictive biomarkers for adverse events during immunotherapy? And are there biomarkers for response to immunotherapy? And for both of these questions, we believe that TCR sequencing plays an important role. We discussed this in the previous webinar, and I think I'm going to elaborate on that point in the coming slides. So now just to the background of the TCR sequencing and the T-cell receptor biology. The T-cell receptor is a heterodimer consisting of an alpha chain and a beta chain. We focus on sequencing the beta chain, which is far more diverse in sequence space and in some respects then more informative. The beta chain is produced by VDJ recombination, where tandemly arranged variable diversity and joining genes recombine to produce a functional receptor. The interface of the VDJ forms the CDR3 sequence, which is incredibly diverse owing to the combinatorial nature of that process, but also the action of exonucleotide chewback at the ends of the VDJ and the addition of non-templated bases by, or also called end additions by TBT. So the CDR3 is so diverse that it can serve as a natural barcode to track T cells over time and measure T cell receptor diversity. With that said, the CDR1 and 2 are also important in determining the antigen specificity of the receptor, and we discussed how the TCR beta LR assay could be used to profile polymorphism within the CDR1 and 2. So now I want to turn to the assay design, and this cartoon shows the design of the LR and SR assays to help you compare the two. The LR assay utilizes primers targeting the framework 1 region of the variable gene and the constant gene region. And this creates an amplicon spanning all 3 CDR regions to enable sequencing of the variable, the germline encoded variable gene as well as the CDR3. And as mentioned in our previous webinar, this allows one to interrogate variable gene polymorphism, which may be a predictive biomarker for adverse events during immunotherapy. Now, owing to the long-amplicon design, the LR assay is not compatible with FFPE preserved materials. And this is where the SR assay plays a complementary role. It's a short-amplicon design targeting the framework 3 region and the joining gene region of the rearranged receptor. The amplicon spans the CDR3 region, which is the crucial region for tracking T-cell clones and measuring diversity. And owing to the short amplicon length, it is compatible with FFPE preserved material, both DNA and RNA. And for reasons that I'll elaborate on in the coming slides, the SR assay is also a great tool for tracking and detection of very rare clones. And that owes in part to the types of sequencing chips that one can use with the SR assay. So to provide you a little bit more detail on this, I want to remind everyone that these assays are designed for the GeneStudio S5 series, of which there are 3 models that differ in their the hardware that is used to process the sequencing data. So there's a difference in the turnaround time and throughput per day. But the other aspect that is important to consider is that with the S5 series, one can use different sequencing chips to obtain different levels of throughput. So going from the 510 chip, which has 2 to 3 million reads output, up to the 550 chip, where one can obtain up to 130 million reads. And there are differences in terms of the read output, but also the supported read lengths. So you'll note that the 520 and 530 chips, for example, support the longest read lengths. And these are really the ideal for the LR assay, which has a long amplicon. Now, the SR assay, owing to its short-amplicon, can be used with all of these chips. So this is where the detection of rare clones comes into play. One can use the highest capacity sequencing chips to obtain very high sequencing coverage of libraries to detect rare clones. And both assays are designed with the philosophy that user hands-on time should be minimal and there should be an end-to-end solution. So these are AmpliSeq protocols with less than 30 minutes hands-on time. You set it up and walk away. The sequencing is very fast. And there's a end-to-end bioinformatics solution on Ion Reporter. And so the total turnaround time, sample to answer, is approximately 48 hours. Now, with respect to the analysis, there's a rich informatics suite that we have developed on Ion Reporter, which is a cloud or server-based analysis platform. This allows one to do things like obtain the sequence and frequency of clones within a sample, but also obtain secondary repertoire features such as T-cell measurements of T-cell expansion or diversity, and readouts of the variable gene usage within a repertoire. I'll also note that the software allows for longitudinal analysis of clones, so tracking clones across multiple samples. So this makes it really nice for evaluating the frequency of a clone of interest in a type of longitudinal experiment. So before showing you data for the SR assay, I want to put everything in perspective. And in this table, I highlight the strengths of both the LR and SR assay, so you can get a sense of when you might want to use one versus the other. Firstly, with input type, as I mentioned, the LR assay is designed for non-FFPE RNA, and it's really the ideal assay for interrogation of peripheral blood. The SR assay can be used with RNA, genomic DNA, and even cell-free DNA, and it's FFPE compatible. And this is in part owing to the short amplicon length of the assay. With respect to the sample barcoding system, the LR assay employs the ion select barcodes, while the TCR beta SR assay utilizes a novel ion dual barcoding system, which I'll talk about in the coming slides. And this barcode system has been designed to help improve the performance of applications where one wants to detect very rare clones and sequence libraries to very high depth. With respect to polymorphism and haplotype analysis of the T-cell receptor beta locus, this is an application that's uniquely enabled by the LR assay, and that's owing to the long-amplicon covering the germline encoded portion of the variable gene. Now, TCR convergence, this is an interesting repertoire feature that may have biomarker utility over a range of applications. This can be evaluated with both the LR and SR assay, owing to the fact that it relies on analysis of the CDR3 region. In terms of assessing T-cell expansion, this is also known as repertoire evenness or clonality. That feature of the repertoire can be evaluated with both assays equally well. Now, as I mentioned, FFPE preserved samples, that's the ideal sample type for the SR assay, and detection and tracking of very rare clones. The SR assay has the advantage owing to the compatibility with our highest capacity chips. So now switching to some data highlighting applications of the SR assay, I'm going to start with looking at FFPE preserved samples, and then we're going to switch to looking at detection of very rare clones. So this slide highlights an experiment to analyze T cells within an FFPE preserved tonsil specimen. And in this experiment, RNA was extracted from the FFPE preserved tonsil to obtain a pool of RNA, of which 25 nanograms was used to prepare one library, and then a separate 25 nanograms to prepare a second library. And I'm highlighting some high-level repertoire features from that analysis, the clones detected, the diversity of the of the repertoire, and then the evenness, which is a measure of T cell expansion. A higher evenness, closer to 1, indicates that there are few expanded clones, while a low evenness indicates that there are 1 or several highly expanded clones in the sample. And you'll note that replicate 1 and 2 are quite similar in terms of the readout of the diversity and evenness. And this is really what you would expect when sequencing material from the same RNA pool. The other thing that one would expect is clonal overlap between these samples, given that they're derived from the same starting material. And indeed, on the right, you'll note that this plot highlighting the frequency of clones and the distribution among the two samples, that there's a significant fraction of those clones that are shared across the two libraries. So that was FFPE RNA. Here's an example from FFPE DNA. And in this example, biopsies were obtained from an individual with colorectal cancer, either from lymph node, a primary tumor, and a liver metastasis. So you'll note first that the lymph node sample revealed a greater number of clones than the primary tumor and liver metastasis. That may be expected based on the biology of the lymph node and the fact that it may accumulate T cells. The other thing that I think is interesting from this analysis is that you will note that there are clones that are found both in the primary tumor and the metastasis. And on the scatter plot on the right, you'll note 34 shared clones. So these shared clones may be at a higher likelihood of being tumor antigen specific, given that they're both infiltrating a tumor and these tumors are at different parts of the body. So this is the type of hypothesis that could be evaluated with downstream studies and highlights the fact that this assay can function well with a small amount of FFPE preserved material. Now, with respect to FFPE experiments, it's also critical to have a method for evaluating the quality of the sample. Because in some cases, the FFPE treatment or the age of the sample will make the sample too degraded to function. And so the molecular biology team has done a great job in developing assays that allow users to readily evaluate the quality of the FFPE RNA or DNA. And here I'm highlighting this CD3 quantification assay, which uses a qPCR of the CD247 gene that's part of the T cell receptor signaling complex. And running this assay on your FFPE preserved materials can allow one to get a sense of whether the material has the possibility of being used for successful library preparation. So that's for RNA, FFPE RNA. There's also an assay for qualifying FFPE preserved DNA samples via the RNase P assay. And the protocols for using these assays are described in the user guide. So I think that's a really nice resource for users of this assay. Now to MRD, or more broadly, detection and tracking of rare T cell clones. This approach utilizes the fact that the CDR3 is incredibly diverse. And it's so diverse that it can be used as a natural barcode to track a clone of interest. And this is actually one of the first applications of NGS-based repertoire sequencing, because using this type of approach, one can readily detect very rare clones that would otherwise be impossible to detect by standard approaches like flow cytometry. And the SR assay is really nice for this, partly because of the ability to generate large libraries and sequence them to great depth using the ion series chips. So just to give you a sense of how this works, typically one will sequence T cells or B cells at a time point where the malignancy is at a high frequency. And by sequencing, one will detect a dominant clone. and that clone will have a CDR3 sequence, which can then be used to track the frequency of that malignancy longitudinally after a treatment has been provided. So after treatment, one asks, what is the frequency of that malignant clone? And if the treatment is very effective, the malignancy may end up being at a very low frequency or even undetectable. So by sequencing, one can search for very rare clones and thus get a sense of whether the malignancy is truly gone or if it's actually at a low level but may return. And there is still some question in the field of what is the limit of detection necessary to determine whether someone is actually has a complete response in terms of this minimal residual disease or along that line. And some people say 1 in 10,000, while it's possible that actually 1 in 100,000 or even 1 in a million may be the most appropriate. So if one wants to detect very rare clones, one has to sequence to great depth and produce large libraries. And this type of application makes the phenomena of barcode bleed-through more of an issue. And this refers to the phenomena where sequences from one sample in a multiplex sequencing run will appear in unrelated samples that are sequenced on the same chip or even in consecutive runs. So in this cartoon example, you could imagine a T-cell malignancy clone in sample 1 could theoretically appear at a low frequency in sample 2, 3, 4, or even samples in a consecutive run. And this is something that one really would like to avoid in these types of applications, because it can potentially confound the interpretation of the results. And so with this in mind, we developed a novel ion dual barcoding kit, which greatly reduces this phenomenon. And the barcode kit utilizes 2 sets of barcode sequences, one at the start of the read and one at the end of the read. And this combination of barcodes allows one to greatly diminish the frequency of these bleed-through events. And as an example, I'm going to show you in this slide an experiment that we performed to test the efficacy of this dual barcoding system to eliminate bleed-through. In this experiment, we created 5 TCR beta sequencing libraries. One that's a mixture of JERCAT and peripheral blood leukocyte cDNA. So there's a single dominating clone within the JERCAT PBL library. And then 4 additional libraries that are created from peripheral blood leukocyte cDNA and thus don't have the JERCAT clone. And we ask, when we sequence these in multiplex, do we find that JERCAT sequence in any of the libraries on the same chip? And at the bottom, you'll note the results of 2 separate experiments where we find that even in cases where the JERCAT clone is at very high frequency in one library, it's not found in the other libraries. So this really is testament to the value of this dual barcoding system for detection and tracking of rare clones. Now, the other aspect of tracking rare clones that I want to bring up is the necessity for construction of large libraries. One needs to construct a large library to increase the chances of detecting a rare clone. And I just want to bring up the fact that when one is preparing libraries from RNA as opposed to DNA, it's easier to create a very large library, owing to the fact that the RNA is already enriched for your target of interest, the T-cell receptor. So in this experiment, this is a table taken from the user guide where we prepared a series of libraries from increasing amounts of cDNA derived from peripheral blood leukocyte, 10 nanograms up to 1 microgram. And these libraries were sequenced to a very high sequencing depth, 30 million reads, and then reads were randomly downsampled. And the table is showing you the number of clones detected. So what you can take from this is that by increasing the input amount of cDNA, one can create very large libraries. Now, for most use cases, we recommend 25 nanograms of cDNA derived from PBL total RNA and sequencing to about 1.5 million reads depth. And you'll note some of the cells in this table are bolded. This is really indicating the point of inflection in the library sequencing where one begins to approach saturation. And so this is kind of a general guidance as to the best point in terms of input amount and sequencing depth to maximize the number of clones detected. So now I'm going to switch to the third section of this talk, T-cell repertoire features of chronic cytomegalovirus infection. And I want to start by describing the biology of cytomegalovirus. Cytomegalovirus is a common and chronic viral infection. In some countries, greater than 95% of the individuals are CMV or cytomegalovirus infection positive. And in the US, for example, approximately 50% of the population has test positive for cytomegalovirus. This is chronic in that it's believed that once one is infected with cytomegalovirus, the infection is never completely eradicated. So it's really a master at immune evasion. And for that reason, firstly, the fact that it's very common. Secondly, that one can readily obtain samples from individuals of known CMV status from blood centers. And third, that the cytomegalovirus has really exquisite mechanisms of immune evasion. It's a really interesting virus to study. And, you know, immune evasion, viral immune evasion may be related or at least provide insight into mechanisms of immune evasion by cancer. So, you know, other aspects of CMV biology that are of interest, it can infect a broad range of cell types, including epithelial cells, which are a common cancer cell type, and also lymphocytes. So even T cells can be infected by cytomegalovirus to some extent. One of the ways in which CMV is thought to evade the immune system is through the generation of CMV-reactive T regulatory cells. I bring this up because it's also thought that T regulatory cells may play a role in cancer immune evasion. And then, you know, another interesting point mentioned in the literature is that CMV infection is associated with large T-cell clonal proliferations, particularly in the elderly. And at the bottom right of the slide, I'm highlighting a figure from a paper looking at CMV-reactive T cells in the elderly. And you'll note the percentage indicating the fraction of cells that were estimated to be CMV-reactive. So it's really, it can be a very profound impact that this virus can have on the repertoire. So with these points, it seems reasonable that one might be able to identify T-cell repertoire features of cytomegalovirus infection. And to do this, we took a look, we generated data and also compared our data to data that was already present in the public domain. So in the coming slides, I'm going to show you data from 3 data sets. Data set 1 is generated using the Oncomine TCR-beta SR assay, using peripheral blood leukocyte total RNA as the input, cDNA from that total RNA, and sequenced on the GeneStudio S5 530 chip to approximately 1.5 million reads depth. And this cDNA was obtained from 35 individuals of known CMV status obtained from the Stanford Blood Center. And this is it's a really easy way to obtain samples of known CMV status. You can purchase a sample from the blood center, and for a small fee, you can obtain the CMV status of that sample. Now, data set 2 is produced using the Oncomine TCR-beta LR assay, using the same set of 35 individuals, again using cDNA derived from total RNA. and again sequenced on the S5 via the 530 chip and targeting 1.5 million reads depth per library. And the third data set is an immunoseq data set from literature. It's a quite large data set consisting of T-cell repertoires from 761 individuals of known CMV status. And this was published in Emerson et al. 2017 Nature Genetics. And this is using gDNA from peripheral blood leukocytes. And this is sequenced using the Illumina HiSeq. So given what I told you about T-cell biology, one could ask whether or not there are differences in T-cell expansion as a function of CMV status. And here I'm showing highlighting an illustration of T-cell evenness. And evenness is a measure of clonal expansion. The lower the evenness, the greater the number of T-cells that are expanded. And evenness is just 1 minus the clonality if you're using immunoseq. And it's also known as the normalized Shannon entropy. So we wanted to know whether evenness differed as a function of CMV status. And here you'll note the results for the TCR beta SR, LR, and the immunoseq data set. And so you'll take a look at the left, and the color indicates the CMV status. And you'll note it's a pretty striking observation that there's a significant difference in TCR evenness as a function of CMV status. CMV positive individuals have a lower evenness, indicating that they tend to have larger clones, expanded clones. And this is true across the 3 data sets. It seems to be a very consistent trend. And you'll note on the right that this trend is so strong that one can actually predict with good accuracy the CMV status exclusively using the evenness of the repertoires. And you'll note on these plots the AUC. This is the area under the curve. The higher the number, the closer to 1, the more accurate or the better the predictor. And so you'll note that that the 3 assays do a good job of discriminating CMV positive and negative based on evenness status. Now, I want to take just a pause and to discuss the significance of this result, because I think it's important for a number of reasons. Firstly, when we talk about T-cell receptor evenness as a predictive biomarker for immunotherapy response. For example, sequencing a repertoire at baseline before someone receives the checkpoint blockade, and then predicting based on that value whether an individual responds. This data suggests that the CMV status has a large impact on evenness. Thus, if one wants to use evenness as a predictive biomarker, one may need to take into account the CMV status of an individual. Otherwise, the effect of the CMV infection may obfuscate differences owing to T-cell responses to tumor. And to my knowledge from the literature, that hasn't been done. And so that's something that I think we can all think about as looking at CMV status and evenness in evaluating the predictive utility of that biomarker. Now, the second aspect is the DNA versus RNA issue. One kind of continuing debate in the repertoire field is what are the advantages or disadvantages of RNA versus DNA? And you'll note that the TCR beta SR and LR assays here were used with RNA template, while the immunoseq was used with DNA template. And they both produce equivalent results, essentially equivalent. So this really emphasizes the notion that RNA can provide a great readout of the repertoire, at least as good, if not better than DNA-based analyses. Other thing to note, it may be evident from the graph I just showed you, but there's a very high correlation in evenness values between the TCR beta SR and LR assays. So irrespective of the assay you use, you can be confident that you're getting an evenness value that is really measuring the biology of those samples. And then the other point I want to mention, there's some discussion in the literature about whether the T cell repertoire ages, or there's some kind of loss of diversity as a function of age. And in these plots, you'll note on the x-axis indicates the age of the donor, and the y-axis indicates the evenness, and the coloring indicates the CMV status. And the lines indicate the best fit for the respective categories. So you'll note that with age, it looks as though in both data sets there tends to be a decrease in evenness, so greater propensity towards expanded clones in the elderly. And that's really consistent with literature. The other point is that in CMV positive individuals, it seems that that this trend is more pronounced. So if one is CMV positive, there is a greater tendency towards large clonal proliferations. And again, this really fits well with what is known in the literature about CMV. Now, I want to turn to convergence. In our previous webinar, we discussed the utility of convergence within the immuno-oncology setting. And the idea is that chronic antigen stimulation may give rise to T cell receptors which have a stereotyped amino acid sequence. And convergent TCRs are those which are identical in amino acid space, but different in nucleotide space. And this fact indicates that T cells independently went through VDJ recombination and proliferated in response to some antigen stimulation. And the one exciting aspect of convergence is that it may preferentially arise owing to chronic antigen stimulation. And we discussed convergence in the context of cancer neoantigens, but one could also imagine that convergent T cell responses might arise following chronic responses to viral antigen. And just as an example, to remind everyone, here's an example of 3 clones which make up a convergent group. They're identical in amino acid space, but different in nucleotide space. So they have the same variable gene, the same CDR3 amino acid sequence, but they're different within the CDR3 nucleotide sequences. And they're different at the VD and DJ junctions, and you'll note that based on the coloring of the text. And I want to point out that owing to the nature of convergence, substitution sequencing errors can readily give rise to artifacts that resemble convergent T cell responses. And you can note that from this slide. And so it's something that this measurement of this feature may be particularly suited to ion torrent sequencing, which has a low level of substitution sequencing errors. And to give you a better idea of why that may be, I just want to remind everyone of the way in which ion torrent sequencing occurs. There's templating of a molecule on a bead, which is placed into a cell that contains a transistor, and when that template is amplified, hydrogen ions are released. And this changes the pH of the well, and that change in pH can be sensed and then translated into sequence. So this sequencing methodology is fast, and it uses sequential flow of nucleotides. So one flow at a time, and that may be one aspect of one reason why this sequencer has a low substitution error rate, because the sequencer really just has to determine whether or not a nucleotide incorporated, not whether or not a nucleotide incorporated and what nucleotide incorporated. So it's kind of a simpler problem to solve in some respects. And here's an example of the substitution error rates using the 530 chip and sequencing the E. coli DH1B genome. On the left, you'll note the types of errors following alignment of those 530 reads to the E. coli genome, and they're broken down by the error mode, substitution, insertion, deletion, and total. So you'll note firstly that that blue line, the substitution sequencing error, is far lower than the others. And just to mention, there's a little, almost like a blip at the very far left around 0 bases. That little uptick in the error rate, to my understanding, is an artifact deriving from the alignment methodology. It's obvious that the error mode is dominated by insertions and deletions. And then moving to the right, you'll note that a comparison of the substitution errors for Illumina, and these error values were taken from a recent paper, Schirmer et al. 2016, using 100 base pair reads. And then for the ion torrent, and with the ion torrent, that value is calculated using read base positions 10 to 100 of the plot on the left. And just a note that the CDR3 sequence, which is the critical part of the T-cell receptor beta library, sits at about 25 bases 25 to 60. So that's at a portion of the read where there's extremely low substitution errors. And the other point to mention is that in all of the substitution error values on the right, this is a combination of substitution errors owing to PCR, so base incorporation errors, and sequencing errors. So it's a combination of those two. So now to return to convergence, we talked about how convergence may be a hallmark of chronic antigen stimulation, and we noted elevated convergence in those who respond to checkpoint blockade immunotherapy. This was discussed in the previous webinar. So it's natural to ask whether convergence is also elevated in CMV positive individuals. And just to mention, this convergent TCR frequency is calculated as the sum of the frequency of clones that are identical in amino acid space but different in nucleotide space. So a variable gene plus CDR3 amino acid shared by 2 or more clones. So applying this definition to the 3 data sets, we note the following with respect to convergence. So I first want to talk about the Oncomine TCR beta assays, and then I'll switch to discussing the immunoseq. You'll note that the TCR beta SR and LR assays do show a tendency towards higher convergence in the CMV positive individuals, suggesting that a subset of the CMV positive individuals may have this tendency towards a response to chronic viral antigen. And to look at that a little bit more closely, I want to bring up the relationship of TCR evenness and convergence, because one thing you could ask is whether or not evenness and convergence are the same, or whether they're actually different features of the repertoire. And you'll note that evenness and convergence are not correlated in the immunoseq data. In the Thermo Fisher data. In the scatter plot on the left, the y-axis indicates TCR evenness, and the x-axis indicates convergence. And you'll note that on the right, convergence values can independently predict CMV status. Evenness values can predict CMV status, and a combination of convergence plus evenness through a logistic regression model can improve the discrimination of CMV status. So now turning to the immunoseq data, when we perform the same analysis of that immunoseq data, the results look different. There appeared to be a very strong correlation between the evenness or clonality and convergence. In this plot, you'll note the correlation of negative 0.94. And one might ask, why is this the case? Hypothetically, what this relationship is saying is that the greater this clone size in a sample, the greater the chance that one will identify a large clone as a convergent clone. And that could be possible if there are residual substitution errors within the data. So errors arising from the PCR or sequencing that are not corrected, that create artifacts resembling convergent TCRs. That's a hypothesis. And to look at this a little bit more closely, we asked whether taking the number of reads per clone within the immunoseq data, the length of the CDR3 per clone, and assuming some level of residual substitution errors, going from a high level of residual substitution error of 1 in 384 down to 1 in 80,000 per base pair, and asking whether such residual substitution error could give rise to the type of data that, the type of values that we observed. And I mentioned 1 in 384 because from recent publications that is suggested as the raw error rate of the Illumina HiSeq, the raw substitution error. And when taking these factors into account, and of course this is a simple model, but we noted that a residual substitution error rate of 1 in 7,500 could give rise to the observed convergence values. So, taken at face value, this would suggest that the residual substitution error rate of the immunoseq data is far lower than the raw substitution rate of the Illumina platform, but is still significant and enough to obfuscate the differences in convergence that may exist between responders and non-responders to immunotherapy or other conditions. And this is obviously an analysis from a simple model, and more insight could be obtained by analysis of the raw data. So I want to finally put everything in perspective with convergence, because I think it's a pretty exciting biomarker. Today we talked about convergence response to viral antigen, and in the previous webinar we discussed convergence in response to tumor neoantigen. And tumor neoantigen can be derived from non-synonymous mutations, which are also interrogated by tumor mutation burden sequencing. They also may derive from aberrant post-translational modifications. Now these are really interesting because they can be highly immunogenic, but they're not detectable by sequencing a tumor. So this is an aspect of tumor immunogenicity that one cannot detect through sequencing a tumor. And I think it's one of the advantages of TCR convergence as a potential biomarker. And then finally, the final antigen source to mention, of course, autoantigens. This convergence could potentially arise in chronic autoimmune disease, of which there's also an aberrant post-translational modification component in many cases. And then the final piece of this, T-cell receptor beta variable gene polymorphism, we discussed in the previous webinar how certain variable gene alleles or sets of alleles, also known as haplotypes, could modulate the way in which T cells interact with HLA, potentially increasing the likelihood of autoantigen recognition. So to summarize what I've told you today, I want to look back at our webinar series. In the first webinar, we discussed how targeted gene expression profiling, tumor mutation burden analysis, and TCR sequencing may be combined to improve understanding of the complex tumor immune cell interactions. In the second webinar, I described how the TCR beta LR assay may allow for assessment of polymorphism and convergence from peripheral blood to enable prediction of adverse events and immunotherapy response. And then here, I discussed how interrogation of FFPE preserved DNA or RNA may be accomplished with the SR assay, which is also great for detecting rare clones. And then the two key points from the analysis of CMV. First, CMV infection strongly shapes the repertoire, and researchers may need to take into account the CMV status when interpreting T-cell repertoire features as potential predictive biomarkers. And then the final point is the sensitivity of TCR convergence measurements to substitution errors And this may be a way in which ion torrent is well suited to this task. So finally, I want to end by saying that I believe that immune repertoire sequencing and the understanding of this data type is really just beginning. And so I'm highly optimistic about the future of this data type in terms of immuno-oncology biomarker research. So with that, I'd like to finish the slide deck and of course take any questions that the audience might have. Are there any questions? I think you might be on mute, by the way, Alexis.
I sincerely apologize about that. You are 100% correct. Thank you, Dr. Looney, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's get started. Our first question is, can this assay be used to detect an adverse immune response to IO treatments?
Great question. The SR assay that I presented today, the short read assay, exclusively covers the CDR3 region. So while it's possible that the CDR3 region alone could be informative to adverse events, what we showed in our previous webinar was that the LR assay, which includes coverage of the variable gene and polymorphism within the variable gene, may provide the data that can be most useful for prediction of adverse events. And this is through interrogation of polymorphism.
Dr. Looney, our next question. Could you comment on DNA versus RNA for FFPE samples? The DNA RNA data in the presentation was from PBL.
Yeah. So I hope I'm going to bring back the slides because I did bring up some data using FFPE preserved samples. Hopefully you can see this. You know, in general, I would say if you have the opportunity to use RNA, I would use RNA because one can more easily create large libraries from a small amount of material. And then secondly, the RNA consists entirely of productive rearrangements. So those that are coding for a T cell receptor that is ultimately expressed. And for that reason, one has in some aspects more efficient sequencing of the T cell repertoire. Whereas from DNA, a fraction of the reads will correspond to unproductive rearrangements that are not expressed.
Could a cancer TCR be used for targeting a CAR T cell?
Could a cancer TCR be used, a T cell, cancer-specific TCR be used for targeting a T cell?
It says for targeting a CAR T cell.
Oh, great question. Can we use this tech? If I understand correctly, can we use TCR sequencing to monitor the activity of therapeutic T cells, CAR T cells? And to that, yes, you can. The only requirement is that the therapeutic T cell has an endogenous T cell receptor that can be amplified and sequenced. And for that application, T cell receptor sequencing is really nice.
We are getting so many great questions, so let's move on to our next one. Are there standard TCR convergence groups defined yet, like for B cell receptor stereotyping? You know,
I don't know the answer to that. I don't know if there are. I think that as the question mentions, alludes to, convergence has been evaluated, I would say, more in B cell receptor repertoires. And that's owing in part to the fact that somatic hypermutation can create very strong signatures of convergence. So I would say that from a historical perspective, there's been more focus on the B-cell repertoire for convergence. But as we've noted today, I think it's also a feature that researchers should look into for T-cell repertoire analysis.
So our next question is, it's number 10, and it says, without dual barcoding, how big of a problem is of bleeding?
Yeah, you know, to my understanding from the data that I've looked at, it's actually quite not a big problem at all. It's really our goal with the ion dual is to strive towards continually improving our product. And part of the reason is that the S5 uses emulsion PCR-based templating. So when one amplifies the template molecules and makes copies of them onto a bead, this occurs within an emulsion bubble. And that emulsion bubble acts as a physical barrier that helps prevent the type of barcode bleed through or index hopping that others have observed in the literature. So in general, given that we use emulsion PCR, that's an aspect that reduces overall reduces the tendency towards barcode bleed through.
Now, Dr. Looney, will molecular tags eliminate all sequencing errors?
Yeah, you know, that's a great question, and one logical question to ask, given that substitution sequencing errors may be critical, how much of those can be eliminated through the use of like molecular tags? And the truth is that molecular tags can, in some cases, reduce errors, but they cannot eliminate all errors because, or they're not likely to, because the molecular tags themselves are subject to PCR and sequencing errors. So there's still some fundamental limit imposed by the library preparation and the sequencing system that is very challenging to overcome. And this is why it's arguably best to avoid the issue of substitution sequencing errors by choosing a platform with low substitution errors to begin with.
Now, our next question is a two-part question. Is it possible to compare data generated by the LR and SR assays? For example, using the LR assay to sequence peripheral blood and the SR to interrogate FFPE preserved tumor from the same individual?
Yes, you can do that. And in fact, the Ion Reporter software is designed for that very thing. So one can certainly evaluate clonal overlap and track clones using both assays. And in the use case the user brought up is really, I think, a common and good choice of the assays to enable getting the most out of your data.
Early in your presentation, biological replicates, sequential biopsies, showed a great difference in TCRB sequences. How does a technical replicate, same cDNA, would look?
Great question. Yeah, technical replicates, as opposed to replicates like that I showed, and I think the slide's highlighted right now. Technical replicates, so resequencing of the same library, will show very high clonal overlap. Greater than 90% of clones will be shared. Now, repertoire sequencing is one of those things where the input material can be incredibly diverse. And so only some of that diversity is captured in a single library, particularly when you're using a small amount of that input material to prepare a library. So for this reason, one does not expect to detect very high clonal overlap when preparing libraries from the same pool of RNA derived from polyclonal T-cell, a polyclonal T-cell population.
Now, it looks like our next question is, do customers have access to the raw data?
Yes, we provide access to all of the raw data, and I think that's a great way for those out there to perform their own experiments and develop their own algorithms if they'd like. And I think that's a great aspect of the system that we developed.
And it looks like we have time for one more question. Have you evaluated TCR convergence in autoimmune diseases such as rheumatoid arthritis?
You know what, we haven't yet, but that's a great question. And rheumatoid arthritis, it's one of those conditions where there's thought to be an important role for aberrant post-translational modification, particularly citrullination. And one could hypothesize that convergence values may be elevated in individuals with rheumatoid arthritis. And given that aspect of the disease and the fact that there is a need for predictive biomarkers, I think that's a great question. That's could be a good use case for the study of convergence.
Thank you again, Dr. Looney. Do you have any final comments for our audience?
I'd like to thank them for taking the time to listen, and I really enjoyed presenting our work, and I wish you the best of luck in your research.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of the registration. If you happen to miss any of our 3-part immuno-oncology series, at the end of this webinar, you will be redirected to the registration page for our first webinar, where you can watch Dr. Simon Cawley highlight recent advancements driven by next-generation sequencing that may lead to a deeper understanding of the cancer immunity cycle. On that page, you will also find a link to the first part of today's presentation, where Dr. Looney describes the advantages of long-amplicon TCRB sequencing via ion torrent for immunotherapy biomarker discovery from peripheral blood. Thank you, Dr. Looney, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand through March of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.